So today we're going to talk about the different types of genetic mutations that you'd find in a cell. But first, I want to review the central dogma of molecular biology and how the genetic information of a cell is stored in the form of DNA, which is then transcribed to form RNA and then translated to generate protein. Nucleotides from the DNA are transcribed to their complementary forms on RNA, which are then read as codons, or groups of 3, to code for specific amino acids in a larger protein. Now, if you mutate one of the nucleotides on DNA, like let's say turning this thymine base into an adenine base, then that will affect the RNA sequence and ultimately the protein that follows. So we say that mutations are mistakes in a cell's DNA that ultimately lead to abnormal protein production. So what are the different types of mutations? Well, the first type of mutations we're going to talk about are called point mutations. Now here I've just written out a random sequence of DNA, which is just a repeating pattern of CTC, which would code for a repeating sequence of GAG in the RNA strand, and finally a protein sequence of 3 glutamate amino acids. So a point mutation is when one of our DNA bases is replaced with another. So in this example, a thymine base is being replaced with an adenine base, which leads to a change in one RNA nucleotide and ultimately a change in one amino acid. Another type of mutation is called frame shift, which works a little differently. So first I'll write out the same DNA, RNA, and protein sequences from before, but now instead of changing one base to another, I'm going to add one to the sequence. And here I've thrown in this extra cytosine base that I've written in blue. Now naturally, this change would lead to an additional guanine base being in the resulting messenger RNA sequence, but what's interesting is that this mutation will change the reading frame of the RNA. Remember that RNA is read in groups of 3, or codons, and being translated to form protein. But now, since we've added an extra G here, all of the codons coming after that extra G will look a little differently. Now instead of having 3 GAG codons, we've swapped out 2 for GGA codons. This means that 2 of our amino acids in the final protein will be changed, and in this example they'll be changed from glutamate to glycine. So you can see that frame shift mutations usually have more significant effects on the final protein than point mutations do. Now it's important to recognize that both of these mutations are classified and named for how they affect the cell's DNA structure, and aren't really named for how they affect the resulting protein. Now our next type of mutations are nonsense mutations and missense mutations. Let's say we have a DNA sequence that normally generates RNA and codes for a cysteine amino acid. A nonsense mutation is any genetic mutation that leads to the RNA sequence becoming a stop codon instead. Now missense mutations are a little different, and they're any genetic mutation that changes an amino acid from one to another. So in this example, our mutation is changing the resulting amino acid from a cysteine to a tryptophan. Now you can see that nonsense mutations probably affect the resulting protein a lot more than missense mutations do, since that new stop codon that we're creating could chop off a huge section of the protein instead of just changing one amino acid to another. So now we can divide the missense mutations even further into a bunch of smaller categories. Silent mutations are when the mutation doesn't actually affect the protein at all. Since many different RNA codons can code for the same amino acid, it's possible that the mutation might not affect the protein at all. So in this example, CCA, CCG, CCT, and CCC in a section of DNA will all end up coding for glycine. So if you change the third base, it wouldn't affect the final protein. Conservative mutations are where the new amino acid is of the same type as the original. So here I have a glutamate and an aspartate, which are both acidic amino acids. So a mutation that swapped out an aspartate for a glutamate would be a conservative mutation. Finally, a non-conservative mutation is one where the new amino acid is of a different type from the original. So here we have a serine amino acid, which is a small polar amino acid, being replaced with phenylalanine, which is a large non-polar aromatic amino acid. And this would be an example of a non-conservative mutation, since serine and phenylalanine are different types of amino acids. Now I'll point out again that all of these mutations are classified and named for how they affect the resulting proteins, and aren't really named for how they affect the cell's DNA. So let's look at a quick example. Sickle cell disease is a disorder where hemoglobin, or HB, which is a protein found in human blood, is mutated into a less active form, which we're going to call HBS. And it results from a single glutamate residue being converted into a valine residue. Now we can classify this mutation as a point mutation since only one DNA base is affected, but we can also say that it's a non-conservative missense mutation since glutamate is being swapped out for valine, and the two are different types of amino acids since glutamate is an acidic amino acid and valine is a non-polar one. So what did we learn? Well, first we learned that mutations originate at the DNA level, but show their effects on the protein level. And second, we learned that we can classify different types of mutations by either their effects on DNA or their effects on protein. In reference to DNA, we have point and frame shift mutations, and in reference to protein, we have missense and nonsense mutations.